you're researching the effects of environmental stimuli and how they affect the growth and development in plants. So before we begin, could you please do a brief introduction of yourself?
Sure. I was born. No, I was grew up in New Orleans. Went to undergraduate work in Spring Hill College, Mobile, Alabama, as a triple major: philosophy, classical languages, and biology. Decided at the end of that to just go on in biology. Got a master's degree at Loyola University, New Orleans, and then went to Yale University for a PhD. Finished up the PhD in biology and decided to do a postdoc there in biophysics, studying membranes. And so I did a couple of years postdoc and then had my first faculty position at the University of Pittsburgh. Was there for 5 years, and then UT made an offer I couldn't refuse, about a 33% increase in salary, and I decided to come here. And I've been here ever since 78.
So why do you like biology?
Well, I think biology is in a relatively early stage of development as a science. I think it's entering a sort of a golden age right now. And the opportunities for discovery are almost unlimited, limited only by your imagination. So I think for people who like sort of being on the frontier, like breakthroughs, like making major discoveries that can change the course of science, I think biology is a great field. A lot of the other fields, I don't know them that well, but I have the impression that they are a little bit more developed, more mature, and so of course every field of science has opportunities for major discoveries, but for me, biology was much more accessible than, say, physics or chemistry, and so jumped in.
Please describe the current research you are working on.
Yes, we are funded by 2 different agencies right now. Well, actually 3. I'm finishing up an NSF grant. National Science Foundation has funded me just about continuously since 1975. I'm switching off from the research that was funded by NSF to similar research being funded by a local entrepreneurial company. So that's one source of funding. And then I have 2 grants from NASA to study the cellular basis of gravity sensing and gravity responding in single cells. So the NSF entrepreneurial research relates to an enzyme called a pyrase that appears to play a major role in regulating plant growth and development. We know that if we knock it out, if we knock out the genes that encode a pyrase, 2 of them, we prevent cells from growing, plant cells. And if we overexpress these proteins, they promote growth and they promote plant productivity. So because of the commercial potential of the NSF work that I had been doing for 30 years or so, this local company felt that they could develop patents and try to develop the commercial potential of that research. So I guess anytime you're talking about an enzyme or a mechanism by which you control plant growth and development, you can imagine that it would have a fair amount of ag potential for feeding the world or helping out in that area. So the NASA research is focused on using a simple single-cell system to study how gravity affects the polarity of that cell. So the cell starts off as a symmetrical sphere, and then under the influence of gravity, it develops a polarization such that the first cell that emerges from it is a root-like cell called a rhizoid that points downward under the influence of gravity. So we are interested in tracking what are the earliest responses of the cell to gravity that lead to the polarization. And so we have been doing that research for a while, and had an opportunity to do an experiment on the space shuttle, and another experiment on this DC-9 airplane that does parabolic flight, called the Vomit Comet, because people get 30 seconds of micro G and sometimes they get disoriented. So we've done experiments in both of those situations on the shuttle and on this DC-9 parabolic flight experiment to show that the early gravity responses of a cell can be modulated by gravity within seconds, very, very rapidly. So we're extremely interested in finding out what are those post-transcriptional, post-translational changes that modify the cell in such a way that it becomes polarized under the influence of gravity. So that's kind of a brief overview of what we do.
And exactly how long have you been working on this?
Yeah, it was interesting when I saw that question. I said, I wonder if I'd have to go back and track the history. And I began doing the work on this enzyme that regulates plant growth and development. Started in 1984. I'm a real slow scientist. It takes me a long time to figure out anything. So, but that work has developed very well, and I've been continuously funded, since that time to study this enzyme. The enzyme is called a pyrase. That's the name of the enzyme. And one of the things this enzyme does is it appears to regulate the concentration of extracellular ATP. As biologists, you know that ATP is the energy currency of the cell, and it is used inside the cell to drive energy-dependent reactions. But it turns out that both animal and plant cells, under certain circumstances, release ATP to the outside, in which case it's called extracellular ATP. And extracellular ATP, both in plants and animals, acts like a hormone. It regulates diverse aspects of human growth and development, and now we know it regulates diverse aspects of plant growth and development. So our interest in this enzyme called a pyrase is that part of its function seems to be to control the concentration of extracellular ATP. The pyrase is an ATPase, and it's actually an NTPase. It can hydrolyze the terminal phosphate off of CTP, UTP, GTP, ATP, and also the terminal phosphate off of the dinucleotides, ADP, GDP, UDP, and so forth. So there's a huge literature on this in animals, about 400 papers published every year, describing the critical role of extracellular nucleotides in regulating human physiology. It's a big, big field. And it's been active since the middle 70s, 1975, I think is when research started. The history of extracellular ATP signaling in plants is a lot more recent. I think you might say really got started in about 2003. And so it's about 12 years old, something like that. Whereas in animals, it's about 50 years old. So we're pioneers in the area of extracellular ATP signaling, and it's been a very fruitful area of research for us. We've enjoyed it. And recently we've found that not only does extracellular ATP modulate plant growth and development in higher plants, but it appears also to play a major role in regulating the gravity response in single cells. So that was an unexpected connection between these two areas of research that we thought were kind of disparate, but they actually came together because this extracellular nucleotide functions appears to be important for both.
So, Dr. Roux, where do you see this research heading?
Well, so let's talk about the gravity research and about the pyrase research. Where I would ideally like to see the gravity research go is we are studying all of our responses in single cells of a fern called Ceratopteris, which is a tropical fern. And it makes spores in the gametophyte generation. And the spores start off as single cells, and under the influence of gravity they develop a polarization when they germinate. And we have shown that there is, in these single cells, under the influence of gravity, a current of calcium that enters the bottom and comes out the top, and that current runs parallel with the vector of gravity. So gravity is pointing down, and this current is moving up. And we found that if we block this current using standard calcium channel blockers, we prevent the gravity response. So since calcium currents are very famous in biology as being critical components of polarity development, both in animals and in plants, like, for example, human eggs. When the sperm hits the egg, there's a calcium current that goes across the egg, and that polarizes the egg. So there's a long history of calcium currents polarizing cells, and makes us think that if the first thing that gravity does to this single cell is develop a calcium current that polarizes the cell, that could be true not only in a fern cell, but also in a human cell. And we know that the human cells that respond to the lack of gravity are human muscle cells and human bone cells. And NASA is very concerned about that, because when astronauts go into space, the longer they spend in time in space, they lose muscle and they lose bone mass. That's a very serious problem, especially if you want to go to Mars, and it's going to take a couple of years to get there and back. So my ideal in the future would be to extrapolate from our research on a fern cell and start using the same techniques that we use to study the fern cell, to study human muscle cells and human bone cells, to see whether they experience the same response to gravity that helps them respond to gravity. And once you know what are the early cellular events that are regulated by gravity, then you can begin to control those and maybe overcome this barrier, which is muscle and bone loss, when people spend a lot of time in space. So there might be a remedy for that if you really understood how cells sense and respond to gravity, not only fern cells, but also human cells. So that would be, for the gravity research, I would see that as, maybe pie in the sky, but an extrapolation from what we're doing to some future advances that would benefit humans. Then on the pyrase research, this company that's currently funding our research would like to see, in the ideal world, in fact, we're going to be doing field trials in next spring to see whether the genetic manipulation of this enzyme can affect crop productivity. And so that would be the immediate benefit of that.
And how do you think your research can help change the world?
Well, I sort of talked about that a little bit already. I think if the gravity research helped facilitate space travel, made it more physiologically feasible. Right now, I'm not sure I want to be the astronaut that would spend 2 years in space and come back without very much bone or muscle. I think that would be a big sacrifice. I mean, if you want to spend the rest of your life on the moon, no problem, because you don't need that much muscle and bone when you're on the moon. There's very little gravity, so it's okay. But if you'd like to come back down to Earth sometime, you might want to have muscle and bone. And I think until we solve that problem, it's going to be a barrier for human exploration of space. So I think that would be one benefit of what we're doing. On the other side, the Epires research, we're really at the early stages of understanding how this enzyme works. And so we think it functions primarily by regulating the concentration of extracellular nucleotides, which regulate plant growth and development. And we have some insights into that, but I think we're at an early stage. And one of the real exciting possibilities for me is that recently we're becoming kind of interested in the possibility that this enzyme also functions in the nucleus. And we have some preliminary data that would suggest that when this Epires enzyme is regulating the NTP concentration in the nucleus, like GTP, UTP, CTP, ATP, that would have a big impact on transcription and other nuclear functions like RNA editing, which there's a lot of chromatin conformation, a lot of activities in the nucleus are dependent on ATP in the nucleus. If it turns out that this Epires enzyme, in addition to functioning to regulate extracellular nucleotides, also regulates nuclear nucleotides, that would have a pretty profound effect, I think, on our expanding our understanding of how DNA is regulated, which I think would be a foundation for biology in general. There's not a whole lot of research on the enzymes that regulate the nucleotide equilibrium in the nucleus, and it's too early for us to predict yet whether the plant Epires does function in the nucleus. But if it does, I would be shocked and surprised if there wasn't also an Epires that functioned in the nucleus in animals and humans. And that would, I think, be a major contribution to just understanding nuclear function, which I think would benefit humans.
Up until now, what is the biggest challenge you have faced in your research, and what is the one thing that helped you overcome this challenge?
I think for me, the challenge of writing both publications and grant proposals that your peers think is worthy of publication in a top journal or being funded. I think for a lot of scientists, the most stressful time is writing the grant proposal and sending it in and waiting for a reply, especially in today's climate where less than 10% of the grants are funded. And also writing a paper that you have spent 3 or 4 years maybe developing the data for, sending it in to have peers just tear it apart and say, well, this is bullshit or something like that. And so you have to develop, I think, two things. One is a high level of self-confidence that no matter what criticisms you get back on your grant proposals or on your publications, you have enough self-confidence to say, I can deal with this. I understand where these criticisms are coming from, and I can overcome them. So I would say the biggest challenge is to stay positive, to stay enthusiastic, despite the critiques that you receive back from your peers. The critiques are welcome, and they really help advance research. I don't mind them, but sometimes they can be, well, for example, if you don't get funded, close up your lab. And so, your career could be over in terms of being able to do research. And I think a lot of scientists here at UT and other places are facing that every year. If the U.S. government continues to fund at 10%, 9%, 8% levels, NIH, NSF, USDA, young people like yourselves can look and say, man, do I want to dedicate my life to entering a gambling deal where I have a 10% chance of making it no matter how good I am? So the motivation has to be in here. It has to be, I'm so driven by the desire to discover, by the desire to understand that no matter what criticism I get, no matter if no one else understands what I'm doing, in a sense, I will try to make what I'm doing more understandable, more relevant, more acceptable to the people that are going to determine whether I can continue doing science. It's a privilege to do science, there's no doubt about it, and I thank the taxpayers every day for that. But from the personal point of view, that's the stress, is to convert what you're doing every day in the lab to a form that convinces others that this is valuable. And it's, I love the challenge. Maybe I'm a masochist, I don't know. I love suffering. No, it's a real challenge. And I think what I would say for young people coming up is, look into your soul. Do you really want to be a discoverer? Do you want to be the first one out there to have an insight that makes a difference in the world? If that drives you, if that motivates you, then you can do it. You just have to be persistent. You have to be self-confident. You have to have a thick skin and say, I'm not going to let, what other people say, some of which is very valid and valuable and some of which is not. And just say, okay, I'm going to deal with that. Go forward.
So what was the main thing that motivated you to start this research?
Again, this notion of discovery, I think I have a personality that likes to be out on the edge, take chances. I feel uncomfortable with safe research. It just gives me the creeps. I figure, it's boring. If somebody else has already done it, I'm just sort of repeating. So I've always enjoyed being a little bit of an oddball, out there on the edge, doing stuff that other people aren't doing stuff that other people think is kind of weird. Maybe like extracellular nucleotides, you got to be kidding me. ATP, cells release ATP to the outside, this valuable commodity. You got to be kidding me. That's not going to happen. Even if it did get out there, it wouldn't last very long. What would it do? Those are questions that I get all the time. I tell people what I'm doing. They say, that's weird, man. Okay, yeah, but guess what? It regulates how plants grow and develop, and it regulates how animals grow and develop. Whether you see it in the textbook or not, that's the reality. And so I think people have different personalities. I like the excitement of being out on the edge. I think unfortunately my sons have inherited that excitement, and one of their birthdays they decided, hey Dad, guess what I'm going to do for my birthday? What's that? My brother and I are going to jump out of a plane. This will be our first time. You got to be kidding me. But anyway.
what's a good way to get involved in research? What would be your advice to students?
Yeah, well, I mean, we have a phenomenal array of brilliant researchers here at UT, and I would say look through the list, pick out something that you're interested in, and go knock on the door and say, hey, I'd really like to be involved in the research that's going on in your lab. And I think, I couldn't say a percentage, but I would guess at least 7 out of 10 of the researchers would welcome you and say, yeah, okay, you're going to have to give up a lot of your time, and you're going to have to be serious about it, and you can't be doing this just to fill an X in a square. You have to do it because you want to do it. But if you fill those criteria and you think you would be motivated to be a pioneer, because virtually, in order to be a faculty member here, you have to be a pioneer. You can't be doing just regular kind of routine vanilla type research. And I think everybody here is doing cutting edge research. And so you've got to be one of those people that wants to do that. Pick out of all the variety of, there's like 70 or 80 biologists that you could choose from. Pick out what you want to do and knock on the door and say, hey, I'd like to be involved. And I'll bet in most cases you will be accepted. Now, don't do that unless you can spend 10, 12 hours a week in addition to your regular schedule of classes to do that. Because the fact of the matter is, if you try to have a research experience, quote unquote, with 3 or 4 hours a week, it ain't going to work. You really have to spend some time on it. You have to fail again and again and again as you try to learn techniques. And if you have that kind of a personality that's willing to go through the challenge, and you're motivated to be a discoverer, then I think the next step is just knock on the door. And the door will be open to you.
Do you have any other advice you would like to give to freshman students pursuing biology?
Yeah, I think, well, this is a general philosophical statement, but know yourself. If you really look into what turns you on, what you really want to do, and follow that path, even if it, you may say, well, I'm not sure I can make a lot of money doing that, or I'm not sure, whatever. But if you know that's what motivates you, then I would say, especially in college now, is to do it, try it, get involved in it, and see how you react to it. You may, like we teach a freshman research initiative class for freshmen, obviously, and it's supposed to introduce them to being able to do novel research. And every year we have maybe 24, 26 students in our class, and we invite as many of them who want to come and do research in the summer after their spring semester is over. And typically 30 to 35%, maybe a third of them say, yeah, I really want to do this. And that's a good batting average. And I think it's good for the others who choose not to go on in research. They have discovered something about themselves. They put their toe in the water and they say, I don't like it. I want to do something else. And that's great. So I think that's one of the values of the Freshman Research Initiative program, is that it allows students to really test the waters and see how do they personally react to actually doing novel research, because it's very frustrating. Most of your experiments are going to fail, but it's the failures. Out of the failures, you learn so much about how to do the next round of experiments and make an advance. And if that process turns you on, then say go for it.
Okay, well, Dr. Roux, thank you so much for your time.
All right, thank you.
And
I enjoyed talking with you.
I wish you luck on what's to come on your research.
Okay, great.